Okay, welcome back. We're going to move forward into protein analysis sequencing using mass spec. I did want to start out just reminding about two-dimensional gel. Here's uh, where they fractionated fractions 1, 2, and 3. And this is the data over here. And it actually discusses the number of spots in each fraction of whatever sample they were looking at. And then bioinformatically, they have tested further. So 23 match the lysate, 33 are in fraction 2, so they're shared, fraction 1 and 2. And there's 156 that are unique. Here's 175, and then here's 127 different proteins. So you see right away, whatever this fractionation scheme is, we're dividing this sample, cell type, whatever it is, and we're getting almost 500 different unique proteins specific for 1 or 3 fractions. And so here is just analysis. Looks like they did some salting out, or not sure what this is, but a comparison of fraction 3 and the original lysate, additional protein spots. So they're talking here was probably the original lysate, and here's protein 3. And you see there are spots in common, and yet there are many spots that are not in common. So again, these would be, if this was wild type and this was a cancer, then you've got multiple proteins to study that are appearing here that are not in the original lysate. So very intriguing. So again, we have native proteins. If they're too big to deal with, we can do trypsinization or trypsin digestion. And each one of those can be mapped on a 2D gel, or it could be through HPLC, or it could be mass spec. You have some type of separation. This is just showing chromatography here, and you're separating the peptides here. If we have 2D gel, here's a spot. We can excise that. They actually have robots that can come in, and this is laid on a grid. And this grid has a position, and that position, a robot comes in and lifts that spot out, digest it, and look at the 4 fractions, and this is their charge to mass ratio. And then you could look in the protein sequence database for matches with the theoretical masses calculated. So I can look in a protein's gene, okay, a gene, and then here is the protein, okay, and then every place that a trypsin could cut, then we could get a fragment. So theoretically, you can take the whole human genome and in silica, in the computer, trypsinate it and get fractions and a protein fingerprint. So now we compare this profile to what would be predicted. And so this what a theoretical mass is calculated for all trypsin release peptides. And now we can figure out what exactly this spot is, just compared using the time of flight, or it's probably the best MADLI time of flight mass spec. So now theoretically, any new virus, bacteria, we get the genome, we can look at say the very virulent strains of these new pathogens, and we can figure out what the proteins are that are in the virulent strains that maybe are not in the less virulent. And virulent means how high level, if it's something's virulent, it's high level to infect and potentially kill. So all this information is coming up right now in the databases. So every causative agent can be analyzed, as well as every cancer from every patient. All that data is coming together. And again, here's the peaks, here's the trypsination, and then the fragments can actually even be figured out as to what amino acids are in each fraction. The mass differences between fractions can construct a partial amino acid sequence, and that allows you to look in the database as well to go and find the gene. So again, one can do massive parallel amounts of this kind of work. So theoretically, every separate protein in 2D gel can be done if you've got enough robotic equipment to do it pretty much all at once. So we're talking high levels of data here. One way to get around some of this is here is proteins that are labeled with different fluors. That's what those are called, fluors. And here is a control, internal standard, and maybe a test protein. So maybe wild type versus cancer. And we do a 2D gel, and we then look in 3 channels and compare the differences here, and look at the different spots and what's different, what's in common, and then you could figure out, like I said, each spot could be excised and the fingerprint determined there. So here's the typical mass spec peptide experiment scheme: cell culture, STS 2-dimensional. Okay, here's and then they're showing a 1D here. That's interesting. Protein, so this should probably be 2D gel. Trypsin, other kinds of digestion. Peptides are separated. You can use HPLC or ion exchange or other chromatography to separate the proteins. Each one has a mass charge. And here is electronic spray, mass spec, or MADLI TOF, so 2 flavors. There's pros and cons for each one. Here's the spray, they're showing that. Here's the peptides, and showing this is the fragments based on charge, which is a mass, which is M/charge, which is Z. And then this is old. Look at this computer. Nobody has a monitor like that anymore. And all sorts of software that can be applied to figure out exactly what this is. So we have the MADLI TOF, which is matrix assisted laser deabsorption ionization time of flight. Wow. And then here's electrospray, mass spec, quadrupole type. So 2 flavors. Proteins are digested with an enzyme to produce a peptide. Peptides are charged. That means they take on an ionized charge, separated according to their different ratios, and each fragment then is then further, each peptide fragment is then further fragmented into ions, and you get a charge mass ratio. And so here's the MADLI TOF, and you have matrix molecules in blue, and here are the molecules, the peptides to be analyzed, and the laser actually releases molecules from the matrix as well as imparts an ionization charge on the fragments here, matrix ion and neutral fractions, and then here are the fragments that have a charge, and they are accelerated because they have different charges through an electrical potential, and the mass spec then catches them and determines how big or small these fragments are. We've covered electrospray to where we've injected a protein mixture, goes through a column, separates out the fragments. Each fragment is mixed with a spray. Here's a spray needle, and each fragment comes out into a droplet, and those are accelerated by this charge here towards the mass spec, and again we get this charge mass ratio detected. So these detectors are then going to computers and measuring exactly what you're getting here. So here is the 2D gel, and you can trypsinate those. You could pick that spot and then trypsinate it if you wanted to, and then you can again digest it, and you can see fragments here. You can do a mass spec, and here's a MADLI mass spec, and here's the intensity, and then the charge mass, and then you can compare to the database here. Okay, so here's the MADLI TOF again. There's a matrix, there's a laser, that's a pulsed UV laser. Notice the charges are imparted onto what you're analyzing, and there are things that are neutral. And they're going through and filter these electric field generators, put all the charged particles in line, and notice you have a large time of flight tube that allows these fragments to separate by size, and then they hit the detector, kind of like throwing a softball versus a golf ball, okay, at a detector. You're going to get fraction of a second difference here. And you can actually even have a sophisticated system where you're bouncing to get even more finer separation. And it looks like they've got some type of floors mixed in for the red, the green, and the blue channel, and figuring out the fractions that way in the MADLI TOF. And the time of flight is discussing this part of it. So this is the MADLI, okay, and this is the time of flight. And together it's MADLI TOF time of flight. Okay, so again, we're detecting that. So you can do electrophoresis. Here's 2D gels. You can digest with trypsin. And then you get the charge, the mass, you get some background, we get the main spectrum here that then you can figure out what exactly it is. Here is cytoskeleton proteomes, and they've got sample A from, I don't know, skeletal muscle from patient 1 versus skeletal muscle patient 2, and they've labeled them with red and blue. And then they've compared the differential expression. And so what's different might be then applied to the mass spec to figure out exactly what spots are in common versus what are different. And so we can do 2 different cell types, wild type and cancer, whatever, and you can compare. And this is just the labeling where you're using doing the same amount of work in one gel, basically. In the blending on the fluorescence, you could have an exact image, and they can be overlaid, and you know exactly what each spot is in common or what spots are different. Here's one that's very high, and this is not so high here. Okay, just one thing that I saw. Yeah, this one is high. See, that's black spot, and it's almost nothing here. So we could take those 2 spots, further digest them, and then do MADLI time of flight or the electron spray, and figured out their mass charge of the fragments there. And here's an actual application here. Get your results faster. This is an advertisement for Symbio Laboratories, and this is Legionella detection. This is Legionella bacteria caused a lung infection, severe lung infection. It was a meeting of the Legionnaires at, I think, Philadelphia. Anyway, there was a water-cooled air conditioner vat that the bacteria were growing in, and the air conditioner sprayed the whole area with the Legionella bacteria. And a lot of these folks were elderly because they had been in the military and whatnot a long time ago, so they were pretty weak anyway. And this caused the first time they detected Legionella as a disease situation. And so now they're showing you can take any sample and use MADLI TOF, and it says same day confirmation result in 7 days. So they can pretty much within the day saying, yeah, we've got Legionella here. It would take 7 to 10 days growing on a petri dish. MADLI TOF, they say they can do it in 4 days. So, and I'm sure this is old, so they probably got it within the day now. But anyway, just let you know, this is very applied work with some of the techniques we've been doing. These are not just theoretical. They actually have very applied work for detection of things. And this is the mass charge is kind of going through there. The ion source, it separates the different fragments, and you have a detector. Here's the MADLI TOF. The matrix is released by a pulse laser. These are filtered in, and you separate by size and the time that these take flight. And this is in a vacuum, so there's no friction, so that doesn't interfere. And these things, kind of like bugs hitting your windshield, pop, pop, pop. And you can figure out the intensity. Is it a swarm of bees hitting you or just one or two? Right, so you can get some intensity. These are protonated peptide molecules because of this laser embedded with these light-absorbing chemicals, the matrix. So that is pulse. They absorb the light. They release that as ionization. The particles that the sample you laid on it are now ionized, and the electric field forces them through a narrow channel and then through the vacuum. And then you have a detector here. Here's just one type, and then notice the wide peaks, and you're saying, well, why don't everybody should use this, right? Look at the narrow, not a lot of cross contamination. See right there, that's two proteins crossing, very clear. This probably costs a lot more. So the poor man's MADLI TOF and the rich folks right there, So when you have this in your house, this is probably a better investment for your MADLI TOF for next Christmas there. In case you need some gifts for the scientists in your life, buy them a MADLI TOF. Sounds like something to drink or something to explode, doesn't it? That's not good. Time of flight. Okay. Oh, look at that. That was neat. And then here's the typical result, these charge mass ratios here, okay, over the intensity. Here's the peptide fingerprinting, same concept, picking the gel, digesting, figuring out the charge mass ratios, and then comparing to the database. So this is identical to what? Each of these fragments is identical to what protein in the database. Here's the same thing, just another figure here. And remember, we can do this all artificially. So we can take the Swissport data protein, and you could predict what it would be by trypsinization. And you could ask the question, is that protein in, say, the mitochondria or the nucleus, if you wanted to. There's probably software that will take any protein sequence and trypsinate it artificially, and you could look at the spectra and you can compare the profiles there in case you wanted to do that. If you had to compare proteins in the nucleus and the mitochondria, you could do that as well. And this may be where I end at this point. So we will pick it up next time and continue our happy, fun time that I know you're all having. Okay, come back.